And then if we zoom in on this structure, the adherence junction, then we come down to figure 1910 in your text, which describes the macromolecular composition of that structure. So no integrin here. The key adhesive protein is an integral membrane protein, single transmembrane span, relatively long extracellular domain, and a relatively short cytoplasmic domain. So reminiscent of focal contacts, but it's different. So the nature of the association, remember, in this case, is a zipper between cells. So that zipper, if you zoom in on it, and had a very powerful magnifying glass, and you could see divalent cations that serve as the cross bridge between those individual cadherin molecules. Those are calcium molecules. So the Ca part of cadherin comes from the calcium, and it would be in this at this level of resolution, it would be right here in this diagram, so that you would have one cadherin from the cell on the left, let's say, being attached to the cadherin on the right, like so, through that bridge. Okay, so the other point about adherence junctions is that, as was the case with focal contacts, you have a bridge between something that's plugged into the membrane and is now contacting another cell, not the extracellular matrix, but is communicating with the outside world in that way, and then there is a plaque that serves as the intermediate between the actin filaments and the extracellular contact. In this case, the actin filaments are not stress fibers. They are forming, or they are the attachment sites of these long circumferential belts that are actually shown on that previous diagram, much nicer. But the idea is that the arrangement of actin filaments in this case is lateral, and if you imagine that going on all along the epithelial surface, then those belts serve as a very strong source of integrity for the epithelial surface, because it goes all the way along thousands and thousands of cells, mediated by those adherence junctions. So that's another important structural role. But remember, we're talking about signaling, so we have to go back to what is actually, what do we need to signal. So to do that, let me draw one more cell just to make the distinction between the types of signalings that would be involved in coming from the focal contacts, depending on where we are in the epithelial surface. So what I'm trying to indicate is that this cell is now completely surrounded by other epithelial cells. So for the system to work appropriately, it needs to survive but not proliferate. But let's imagine this cell or this cell is at a boundary that needs to be filled in, or a zone that needs to be filled in. So this cell needs to survive, but it also needs to proliferate. So the signaling has got to be robust enough to handle two completely different types of signals. There is a very big difference between surviving and proliferating. Anybody that has cancer will know that. So how is it that these adherence junctions can achieve both of those types of signaling? So as it turns out, it's going to involve the very important role of one of the proteins in the plaque associated with cadherence junctions. So at this level of resolution, we can just imagine that as blue macromolecular complexes that are in contact with cadherens on the lateral surface of the plasma membrane, and then are attached to actin in the cytosol down here. So in the form of a plaque, beta-catenin is, you should think of, trapped in the surface of the membrane and not in the cytoplasm. And so what you say, well, the importance of that fact is that there's a nucleus, and the nucleus needs to be told when to signal proliferation. So that signal has got to come from somewhere, and it turns out that beta-catenin is one of the key signal molecules that if it gets to the nucleus, turns on genes that are associated with progression through the cell cycle. So before we go any further, we can just indicate in very simple terms where the beta-catenin is going to be in these cells, because there are plaques all around this cell. Then you can imagine all the beta-catenin is going to be here and here. But this cell has fewer plaques as a result of the fact that it is not completely enclosed. It doesn't have as many. So in that case, beta-catenin is going to be much more free to roam about the cabin. And if that happens, then some of that beta-catenin can migrate into the nucleus, where it then triggers the cell cycle. So in this real simple case, it's just a physical restriction of the location of beta-catenin that means the difference between survive and proliferate. The fact that in the case of the cells that have more of the plaques, because they have more adherens junctions, because they are attached by more cells, they have a diluted signal that would trigger the cell cycle. And that is a major contributor in terms of the overall structural integrity of the epithelial surface, and that is a major means by which the signaling coming from the side that could either say survive or not is achieved. But there is another involvement of the plaques and beta-catenin that is associated with the cell cycle and some of the things that we're going to be talking about next that I wanted to just mention briefly. So let's go back to a diagram that involves the plaque, and remember that beta-catenin is going to be one of the proteins that are contained within it if there is an adherens junction. So there is in fact an additional component to this story that I'm not going to go into in any detail, but I did want to mention it just because I didn't want it to go completely unmentioned. And that stems from the fact that in reality, if you have beta-catenin here, it doesn't necessarily mean that this cell is destined to proliferate, to undergo another round of the cell cycle. And that is because there is a very, very important regulatory mechanism that operates on it, and it involves two of our old friends, so I kind of wanted to at least mention that. The overall structure involves a very important protein called APC. It's actually named after the cancer that is the susceptibility if you have mutations in APC. I'm not going to go into that. I'm hoping that you've heard about it in another, like a cancer biology class. But another subunit of that protein, or the complex, is glycogen synthetase kinase 3. So let's say it's right here. So in the situation in which a cell is supposed to be instructed to survive but not proliferate, the GSK3 is going to be active, and one of the things that it does, one of the many things that it does, is phosphorylate beta-catenin when it's in complex with APC. And that phosphorylation is a signal for E3 ligases to ubiquitinate beta-catenin and send it to the proteasome. So even though some of the beta-catenin is not bound up in plaques in this cell, what's left is being actively removed from the cytoplasm before it can get to the nucleus and trigger the cell cycle. So this cell would be surviving. But the importance of this mechanism is that the APC-based mechanism is that it can be itself regulated. And there's a very important regulatory process, important in development especially, that I've alluded to, but I wanted to mention just in passing now. that involves the developmental signal Wnt. And this is a G protein coupled receptor pathway that is not too complicated, but I don't want to go into it other than to say that what happens as a result of the appearance of Wnt as an extracellular signal is that it's recognized by a G protein coupled receptor type receptor, and that ultimately leads to the inactivation of GSK3, which of course then prevents the phosphorylation of beta-catenin, which of course prevents its ubiquitination, which means that it goes to the nucleus, which means the cell cycle kicks in. So Wnt is a major cell proliferation signal, and the way it works is ultimately through the same direct signal that is beta-catenin in the nucleus, leading to the activation of genes associated with the cell cycle that we associate with the presence or absence of adherens junctions, and the overall effect that has on the cytoplasmic concentration of beta-catenin, and therefore indirectly the nuclear concentration of beta-catenin.